does a world without Parkinson, diabetes, Alzheimer's, or even aging sound? Well, thanks to stem cell technology, all of this may be possible soon. Hi, my name is Priyanka Jain, and I'll be talking to you today about stem cells. Let's start with a thought-provoking question. If normal cells die within days or weeks, how are we able to live for decades on end? Well, the answer lies within the fundamental definition of what a stem cell is. It is the reason we are able to outlive our individual cells. So hold on, what does a stem cell do? A stem cell is able to divide itself and differentiate itself indefinitely, unlike other cells. Additionally, it can transform into other specialized cells, such as skin, blood, or muscle cells. Let's look at an analogy to better understand this topic. Essentially, a stem cell is a stem that is able to produce all the different kinds of cells that the body needs. They are blank slates that can become anything. But here's where it gets complicated. There are two different types of stem cells, adult and embryonic stem cells. Adult stem cells can only transform into cells of a specific pathway. For example, blood stem cells can only transform into cells of the circulatory system, such as plasma, red, or white blood cells, but not muscle cells, for instance. Embryonic stem cells can transform into any type of cell that they want, including muscle, nerve, or even brain cells. This term is called pluripotent. What makes stem cell research so hyped is the potential that embryonic stem cells hold. You can first take a stem cell and then multiply it into large numbers, and then you can change it into any type of cell you want and be able to deliver it to a damaged organ or system in large quantities. But where would we obtain these cells? They are called induced pluripotent stem cells, or iPS for short. Let's look at some background info. So the genes in our nucleus give the cell its identity. There are 23 pairs of chromosomes made of long strands of DNA found in the nucleus. And these chromosomes are divided into regions called genes that direct the cell to make a particular protein. All cells have the same genes, but liver cells, for example, only have the genes to make liver proteins turned on, due to unwound open areas on the DNA coils. All other genes to make hard skin or embryonic pluripotent stem cells are turned off due to tightly wound DNA. So then a powerful question was asked. Can we unwind the DNA coil and make the specialized cell revert back to an embryonic stem cell? It was found that in the presence of 4 proteins, the DNA can unwind and turn back the clock to become a fully functioning embryonic stem cell. So now a skin cell can be converted back into an embryonic stem cell, which can then be specialized into any different type of cell we want. So the holy grail of stem cell technology now lies in being able to actually control how to specialize these embryonic stem cells. This opens a door to revolutionizing human medicine.